In the name of Allah, who is most beneficent and merciful, my name is Umaybiba. My ID is 014. I'm from Chemistry Department. And today, topic of my presentation is lipase and alpha-amylase. First of all, I'll tell you about contents of my presentation, which include introduction of lipase, synthesis, enzymatic activity of lipase and its applications. Then I'll introduce alpha-amylase, its synthesis, its biological reactions, and at last but not least, applications of alpha-amylase. So let's start with introduction of lipase. Lipase is an enzyme that breaks down triglycerides into free fatty acids and glycerol. Lipases are present in pancreatic secretions and are responsible for fat digestion. There are many different types of lipases. For example, hepatic lipases are in liver, hormone-sensitive lipases in adipocytes, lipoprotein lipase in vascular endothelial surface, and pancreatic lipase in the small intestine. Here you can see basic information about chemistry of lipase. The structure you can see molecular formula is C45A69NO8. Molecular weight is 752.0 and the optimum pH is 7.0. So now you can see synthesis by fermentation. How it can be synthesized by fermentation. It can be synthesized by fermentation by the solid state fermentation of soybean meal, and the substrate is obtained from a local soybean oil industry. The meal is the residue of soybean oil extraction after cold press and solvent extraction. The meal is sieved, and the three major fractions based on the particle diameter were 1 to 2 millimeter, 0.5 to 1 millimeter, and 0.25 to 0.50 millimeter. So the 0.5 to 1 millimeter fraction is collected and stored at 18 degrees Celsius temperature until the moment of utilization. Fermentation is carried out with 10 grams of dry meal in cylindrical tray reactors covered with hydrophobic fabric. The moisture content of the meal is adjusted to 55 milliliter per 100 gram. So the different supplements are then added. The effect of different nitrogen and carbon sources are evaluated in terms of lipase production using the two microorganisms which are selected before fermentation. Different supplements are added to soybean meal because they are added as substrate, and the respective C/N ratios means carbon over nitrogen ratios are being noted. All supplements are added at 1 gram per 100 gram concentration except when stated. This is the table in which you can observe C/N ratio in different components. So when there is soybean oil with the soybean meal, then the C/N ratio is 6.23. When there is sugarcane molasses with soybean meal, the C/N ratio is 6.15. When there is corn starch liquor, then the C/N ratio is 6.11. In the soybean meal plus yeast hydrolysate, the C/N ratio is 6.09. In pure soybean meal, C/N ratio is 6.11. When urea is added to soybean meal, C/N ratio is 5.76. And when urea and soybean oil both are added to soybean meal, then C/N ratio is 6.11. After sterilization, each reactor is inoculated with a suspension of spores to a final count of 10 spores per gram and inoculated in climatic chamber at a temperature of 27 degrees Celsius with a humid air injection. After 48 hours of fermentation, phosphate buffer at a proportion of 4:1 is added to fermented cake, and the enzyme extraction is carried out in an orbital shaker at 150 RPM, 35 degrees Celsius for 30 minutes. So these are the specific conditions in this type of fermentation. Then we'll discuss the measurement of activity of lipase. So how it is measured. 1 unit of lipase activity is defined as the amount of enzyme preparation necessary to produce 1 micromole of free acid per minute in the assay conditions. The results are expressed in terms of units per gram of dry substrate. Then enzymatic activity. Humans also have several related enzymes, including hepatic lipase, endothelial lipase, and lipoprotein lipase. So working of lipase at different locations in a human is given. I'll discuss them one by one. Bile salt dependent lipase. It is present in pancreas, our breast milk, and what is working? It aids in the digestion of fats. Second one is pancreatic lipase. It is present in digestive juice, and its working is basically in order to exhibit optimal enzyme activity in the gut lumen. PL requires, PL stands for pancreatic lipase, and it requires another protein called lipase, which is also created by the pancreas. Then there is lysosomal lipase, which is present in interior space of organelle lysosome, also referred to as lysosomal acid lipase or acid cholesterol ester hydrolase. Then hepatic lipase, which is present in endothelium. So what is its working? Hepatic lipase acts on the remaining lipids carried on lipoproteins in the blood to degrade, to regenerate LDL, low density lipoprotein. Next is lipoprotein lipase. It is present in endothelium. Lipoprotein lipase functions in the blood to act on triacylglycerides carried on VLDL, very low density lipoprotein, so that cells can take up the freed fatty acids. Then gastric lipase, and after that pancreatic lipase related protein. So gastric lipase is present in digestive juice, and pancreatic lipase related protein 1 is also present in digestive juice, but their workings are different. Gastric lipase works, it functions in the infant at a near neutral pH to aid in the digestion of the lipids, but below 1, pancreatic lipase related protein 1 is very similar to PLRP2 and PL by amino acid sequence. All 3 genes probably arose via gene duplication of a single. However, PLRP1 is devoid of detectable lipase activity and its function remains unknown even though it is conserved in other mammals. So last one is lingual lipase, which is present in saliva. It is active at gastric pH levels. Optimum pH for this enzyme is about 3.5 to 6, and it is secreted by several of the salivary glands. Here you can see applications of lipase in food industry for aroma and flavor enhancement, synthesis of natural dyes, synthesis of flavors, and emulsifiers. In paper, textile, and leather industry for removal of hydrophobic substances. In environmental bioremediation for treatment of effluents and industrial waste. In pharmaceutical and medical industry for separation of enantiomers and for production of biosensors. In biofuels for biodiesel production. In chemical industries for ester synthesis for production of detergent and emulsifiers and for production of polymers. So its applications are not limited. It is very significant in all fields of science. Now we'll discuss about alpha-amylase. It is an enzyme that hydrolyzes alpha bonds of large alpha-linked polysaccharides such as starch and glycogen, yielding shorter chain thereof dextrins and maltose. It is the major form of amylase, which is found in humans and other type of mammals. First of all, you can see chemical structure of lipase. Its molecular formula is C9H14N4O3. Optimum pH is 7.0. So now I'll discuss its synthesis by fermentation. Fungal alpha amylase is produced by submerged fermentation of aspergillus, followed by purification and formulation. The product depolymerizes starch to produce oligosaccharides and a small amount of dextrin. The product performs well in flour correction and baking industry. Moving towards the mechanism, fungal alpha amylase is type of alpha amylase that hydrolyzes the alpha 1,4 glucosidic linkages of gelatinized starch and soluble dextrin randomly, giving rise to oligosaccharides and a small amount of dextrin, which is beneficial for flour correction, yeast growth, and crumb structure, as well as volume of baked products. Now we'll discuss reaction parameters. Activity temperature should be in range of 45 degrees Celsius to 65 degrees Celsius. Optimum temperature should be in range of 50 degrees Celsius to 65 degrees Celsius. The range of activity pH should be 4.5 to 6.0, and the range of optimum pH should be 4.8 to 5.5. So what should be the product parameters which are formed by this kind of fermentation? The physical form should be in powder, color should be light blue, and odor should be normal microbial fermentation. So what's about package and storage of the product? Package, it can be packaged in 25 kgs per drum or 1,125 kgs per drum. Storage, keep sealed in a dry and cool place and avoids direct sunlight. Its shelf life is 12 months in a dry and cool place. So how its activity is measured? 1 unit of fungal alpha amylase equals to the amount of enzyme which hydrolyzes soluble starch to get 10 milligram, reducing sugar at 40 degrees Celsius and pH 5.0 in 30 minutes. Next, there are its biological reactions. First one is breakage of starch by saliva amylase. How starch is break down with the help of the alpha amylase. In this reaction, you can see it is being broken in the dextrins and in monomer of the glucose. The second one is activity of pancreatic amylase. Pancreatic alpha amylase randomly cleaves the alpha glycosidic linkages of amylose to yield dextrin, maltose, or maltotriose. It adopts a double displacement mechanism with retention of anomeric configuration. In humans, the salivary amylase evolved from copy of it. In this reaction, you can see starch in form of amylose. It is being degraded and converted into maltose or shorter polysaccharides with the help of the pancreatic amylase. So these are the two basic biological reactions of alpha amylase. Then we discuss about what are applications of alpha amylase in industries and in medical use. Alpha amylase is used in ethanol production to break starches in grains into fermentable sugars. The first step in the production of high fructose corn syrup is the treatment of corn starch with alpha amylase, producing shorter chains of sugar oligosaccharides, and it is also used in some detergents, especially dishwashing and starch removing detergents. What is its medical use? It has exhibited efficiency in degrading polymicrobial bacterial biofilms by hydrolyzing the alpha glycosidic linkages with the structural metric exopolysaccharides of the extracellular polymeric substances. So these are the applications of alpha amylase. These are the references from which I have collected material for my presentation. So thank you.